All right, looking at how biotechnology is used to treat disease, we can use that recombinant DNA technology that we talked about with agriculture, but to make medications. An example of that would be insulin. So insulin historically would come from slaughterhouses where the animals were being slaughtered for their meat. The pancreas would be saved from the cattle and pigs and shipped to a different location where they would then grind up the pig and cattle pancreas and extract and purify that animal insulin. Problem with that was it was very expensive. It would take like 300 pounds of pig pancreas to make just 1 pound of the insulin out of it. And another problem with that, people who were taking this insulin from cattle or pigs could have allergic reactions to it because it wasn't human insulin. They could be allergic to the pig or the cattle version of the insulin. So it's incredibly expensive, and a lot of people would have allergic reactions in addition to being diabetic. In 1982, that was the first time a transgenic medication was approved by the FDA, and what they did was looked into the human genome and found the piece of DNA that makes insulin and its regulatory region. They cut that out using restriction enzymes. They put it into a plasmid, so now we've got this small circle of DNA with the insulin gene and all of its regulatory region. And they inserted that into E. coli bacteria. So now we have the E. coli that produces human insulin. We allow the E. coli to grow in huge tanks, and once it's grown enough and produced enough insulin, then we drain the tanks, take out all of the E. coli, break open their cells, and purify that human insulin. So now it's less expensive because we can produce it in larger quantities, and it's human insulin, so people taking it do not have allergic reactions. And that's just one example. Human growth hormone has also been produced this way. Other medications to treat anemia and hemophilia, and the list goes on and on and on. And in fact, about 98% of patents that have been approved or applied for in the last 3 years for new medications have all been produced using some type of transgene. Today, there are over 1,500 biotech companies, and it produces over $40 billion in revenue every year. And in fact, Iowa is kind of the center of that. So you hear on the news a lot about kind of electronic companies, so Apple and places like that opening companies here in Iowa. Well, the same is true for biotech. Biotech is kind of centered here in Iowa because we have the resources and the workforce, so there's an increasing number of biotech companies in the area, and a very good way to become employed. If you're interested in biology at all, you could be successful in biotechnology. Biotech has been used to diagnose and also prevent and cure diseases sometimes. So there's a particular disease called severe combined immunodeficiency disease, and that's also known as the bubble boy syndrome. Not that awful movie that was produced a couple years ago, but his actual syndrome. This young man was born without an immune system, and he lived in this plastic tubing his entire life. So everything he ever came in contact with had to be sterilized because he didn't have an immune system. Basically, he was born without it. So all of his food, all of his toys, everything had to be sterilized before it was transferred into his area where he lived. And at one point, bacteria got in, and he actually got sick from it and died because his body simply had no way of fighting it off. And in the 90s, some people with the exact same syndrome were completely cured of it. They were cured because your immune system comes from your bone marrow. So your bone marrow produces your white blood cells, which are your immune system cells, and your red blood cells. What they did to cure these individuals is they extracted some of their bone marrow, so they used a really big needle on a syringe and extracted their bone marrow, and they grew that in a little petri dish, and they treated those individual cells with DNA to replace the genes that were not functioning properly. So they treated their bone marrow cells with functional DNA, and then they put that back into the patients. So some people were completely cured, so it fixed their severe combined immunodeficiency disease. The problem with that technique is it's not 100% effective. Sometimes they might cure the bone marrow disease, but cause other problems, such as cancer. The reason behind that is right now we can add DNA, but we can't tell exactly where it's going to go. So here's our DNA. And maybe in this area right here, it would be okay to put a gene in because this is non-functional DNA. It doesn't make a protein. But over here, perhaps there is an important region that makes a protein. Now, if when we add the DNA to people's genome, if it jumps into this area, so we add the new gene here, then we'll just cure them and not cause a problem. However, if it jumps in over in the protein, we'll disrupt their protein. If it jumps in a regulatory area, we could cause problems, or maybe it could jump in someplace else that regulates cell division. So if we interrupt cell division processes, that leads to cancer. We can interrupt proteins. We simply can't control where that DNA inserts itself yet. So that's why using biotech on humans is only kind of last resort when there's something incredibly severe that would be deadly anyway, because there are a lot of potential risks. It could just by chance jump in someplace that's just fine, but there's always that possibility of that DNA accidentally going someplace else. There are currently over 500 different gene therapies that are being used, but again, no clear success because of that high risk factor of manipulating it and causing problems with other proteins, causing problems with cell division, leading to cancer. So there's lots of issues. So also, depending on the condition, bone marrow is easy to do because we can extract the bone marrow, treat individual cells outside the body, and then return them to the body. But what if it's your heart? We can't just extract your heart and treat it and return it. So treating specific cells in the body, if you can't remove the cells, that's a problem. How do you treat enough of the cells to produce the effect that you're looking for? If you find some way to deliver it to the circulatory system, maybe, how do you make sure it doesn't have side effects in other cells? And again, making sure that your genes get into the target cell. You wouldn't want to try curing diabetes by putting insulin-producing cells in anything else other than the pancreas. So there's lots of problems or concerns that come along with treating using DNA. You could also get diagnosis. So right now the most common use of biotechnology as far as prevention is diagnosing. So if two people know that they're both carrying genes for awful diseases, maybe like Tay-Sachs, it's lethal, cystic fibrosis, which can also be deadly, sickle cell anemia, they might choose to go to a genetic counselor, talk to them, know their risk factors, and then go through something like in vitro fertilization, where they collect eggs and sperm from the parents, combine them in a lab environment, and then test each of those embryos and only return the ones that don't have Tay-Sachs or cystic fibrosis to the mother to grow into a baby. You can also, if you're expecting already, you can test before birth using amniocentesis. That's where you basically take a large needle and insert that into the abdominal area where the baby is and extract some of the fluid from around the baby, and then you can test that fluid for individual cells because the baby's losing skin cells and stuff like that even in the womb, so that fluid would contain cells from the baby, so you could test for different genetic conditions. That's used a lot to make sure you're medically prepared. Maybe a child has Down syndrome. Well, not only do they have kind of the classic things with Down syndrome, but Down syndrome individuals often have heart defects and other things. You may want to have additional medical personnel available when the baby's delivered. Chronic villi sampling is similar. It gets cells from the baby, and it's slightly less invasive than this large needle in through the abdomen. You could look for disease genes in healthy individuals, so screening people to see if they carry breast cancer genes. So Angelina Jolie has been in the news for having a double mastectomy, removing both of her breasts because she carries a gene for breast cancer. In fact, her aunt died of the disease as well. And the problem with that, though, if you get tested for a genetic disease, do you really want to know? And who else should have access to that information? Because once you know that you have a particular disease, your health insurance provider should be informed of it. If you know, I mean, you're obligated to tell them. Should they really have access to that? There is a law in effect called the Genetic Information Non-Discrimination Act. It was passed in 2008. That act says, well, they still have to insure you. Doesn't say anything about what rates they can charge, but that does not cover life insurance, disability, or long-term care. What if you have a disease like Huntington's disease? Huntington's disease is a late onset, lethal condition where it usually occurs in men between the ages of maybe 45 and 65, depending on kind of what the family history is. And in Huntington's disease, everything is normal your entire life until that disease starts, again, after usually age 45, and then it gradually breaks down your brain tissue. So you slowly just start to forget things, you can't remember things, and eventually you get to the point where you can't walk, you can't talk, you can't feed yourself or take care of your own personal hygiene, and you become an adult infant where other people have to care for you. Well, if you get tested genetically to see if you have Huntington's disease, it's dominant. So if you've done those Punnett squares in class, if you have the gene for Huntington's disease, you're going to develop Huntington's disease. Your insurance company, if they know that, they can deny you life insurance. Well, you know you're going to die. They can deny you disability insurance. You're going to need that. And they can deny you long-term care insurance. So genetic discrimination is a huge problem. If you get tested and know that information, how do you then protect it? And would you really want to know that you have some of those diseases? Would that make you live your life differently? Would that be better or worse? So there's a lot of questions that go along with some of these diagnoses, and should we use them or not? We can use it to prevent disease. So I've already mentioned in vitro fertilization with things like Tay-Sachs, prenatal diagnosis, that's using the amniocentesis or the chronic villi sampling for like cystic fibrosis, Down syndrome, and our Genetic Non-Discrimination Act. Again, there's lots and lots of moral issues. What's the right thing to do with that information? Another type of cloning, another type of use for, I guess, biotechnology is cloning. So reproductive cloning. Dolly was born in 1997, and this was an experiment done in Scotland by Ian Wilmot. And what he did was take an adult cell from a sheep, and he took a second sheep and removed the nucleus of the egg. And then he combined this adult cell and the egg cell, a little bit of electricity to start it dividing, and so now we have this adult DNA from this dark colored sheep becoming a new individual. And this has gotten better over time, but it was fairly imprecise. They tried this 272 times, and they managed to get one baby sheep full term to get through this whole process. And that one was Dolly, and that was the first successful cloning of a mammal, which was scary because humans are mammals. Now that we know we can produce Dolly, we can technically clone a human. Dolly died in 2003, so she lived about 6 years or so. The interesting thing about Dolly is at the end of all chromosomes are something called telomeres. And telomeres, we've talked about before, they shorten just a little bit each time the cells divide. Because Dolly was produced from adult DNA, her telomeres had already aged 6 years before she was born. So basically, she started with reduced telomeres. And by the age of 6, she was suffering from a lot of issues that usually only old sheep get, because sheep live to be about 12 years old. So her shortened telomeres also shortened her own lifespan, although she was able to have her own offspring fairly normally. So it had mostly an effect on her lifespan. An interesting thing about identical twins in humans, they are a natural type of clone. It's just one egg that splits during development. But cloning humans on purpose, without the kind of accidental identical twins, has some huge ethical issues. Who should be cloned? Their lifespan is going to be reduced depending on how old the original adult was. Would they have the same rights as other individuals, or would the clones be treated differently? So we simply don't know. There's lots of ethical issues that go along with potentially cloning human beings. Oh, also I should mention, Dolly got her name because the original cell that came from the adult was from an udder. And an udder, if you compare the anatomy to a human, the udder is the equivalent body part as the human breast. And there's a famous singer, very well known for the size of her bosom, and that would be Dolly Parton. And so the scientists there in Scotland decided to name this clone sheep Dolly because she originally grew from a breast cell. So that's why she was named Dolly. Cloning is a potential way that we could perhaps preserve endangered species. So there was a Siberian ibex that was cloned. But there's lots of issues with that. If we clone them and bring them back, do we have habitat for those species? So they have gone extinct mostly because of habitat loss due to human activities. So is it right to then bring them back if they have no habitat to survive in? Could we do something like Jurassic Park and bring back the dinosaurs? No, that would be highly unlikely. DNA is very stable, but it does degrade over time. About the oldest DNA that we've managed to get reliable sequence from is from a Neanderthal that's 30,000 years old. And since dinosaurs are 65 million years old, there's just no DNA left in there. Also, you have to have a compatible mother to carry the offspring. So the ibex that was cloned, they used a closely related goat species for the baby ibex, but there's really no compatible mother around to carry a baby dino, or egg that would allow a baby dino to grow in it. Could try crocodiles or other types of large reptiles or lizards, but there's a lot of cell communication that goes on between a mother and the offspring, and also within the egg. And if the egg proteins don't recognize it as the same species, they'll attack it and try to kill it off. So you need to have a mother that's compatible, and that's unrealistic when we're talking about T-Rex. So lots of animals have been cloned since '97. They include horses, mules, cows, pigs, cats, mice, and goats. Some of them were transgenic, so they produced medication. So there's a type of goat over here, and they produce spider silk in their milk. And you have to genetically engineer them and clone them to get more goats that produce the spider silk. That's important in treating burns. So victims of severe burns, they take the spider silk in the large quantities from the goat milk, and they purify that and weave it into basically bandages to wrap around the burn victim's wounds, because if you use cotton, that sticks to the burn and causes pain and issues. And the spider silk, if you get it in large enough quantities, it protects the burned area. It doesn't cause pain, doesn't stick to the wound, prevents infections, things like that. Other medications have come from different animals. Oftentimes it's expressed in their milk, so you don't have to harm the animal at all. You just harvest the milk and purify the medication for treating things like cystic fibrosis. Cats have been cloned. The first cloned cat was named Copycat, so after its original version. So the use of embryonic stem cells is another possible way of using biotechnology, and we're going to talk about that, but let's first look at what is an embryonic stem cell. So an embryonic stem cell are cells that occur very, very early during development, and they can develop into any type of tissue. So as the blastocyst continues to develop, they start to divert and develop into heart tissue and lung tissue and muscle tissue and nerve tissue. And so this very early blastocyst is after about 5 days. What happens in reproduction, the ovaries produce the eggs, and they release usually just one egg each cycle, and those eggs are pulled across into the fallopian tube. So this is the fallopian tube in the womb down here. Fertilization actually occurs up in the fallopian tube. So the eggs are produced and jump across, and then they're fertilized while they're in the fallopian tube. So once fertilization occurs, that's where we have our zygote. So the sperm and egg have combined, so that's our very first cell. And that zygote starts to divide. So day 1, day 2, 3, at about day 4 to 5 here, it's still traveling through the fallopian tubes, and that's the point where it's a blastocyst. And it looks like this. It's just a ball of cells that's hollow, and it has a few cells stuck to the inside of it. It's these cells that will eventually grow and form the fetus, and then this ball of cells on the outside will form kind of the embryonic sac, so surrounding the baby, protecting it. This is the stage where you need to get embryonic stem cells before they've divided into anything else. After day 5, continues traveling, and about day 7, then the egg has implanted in the uterine wall, started to grow a blood connection to the mother, so now it's getting its nutrition from the mother, and is an actual embryo after day 7. But prior to that, it's really just a collection of cells and hasn't formed anything, still doesn't have a heartbeat or any recognizable parts. It's just a ball of cells. So that's what an embryonic stem cell is. And different religions have different definitions of exactly when life occurs, ranging all the way from fertilization up through when the baby takes its first breath of air. So depending on what your backgrounds are and your beliefs, the scale of life is somewhere along here or even after birth. So that's what an embryonic stem cell is. But you need to get them at this day 4 and 5 before it's even implanted in the mother, when it's just a ball of cells. Adult stem cells have been used for treating human diseases, but they're not as versatile. So they can make many types of cells, but not all kinds of cells. Because when we start with the blastocyst, it can make pretty much any type. But once you diverge into one of these lines, then you're limited in what types of cells you can make. If nerve cells are over here and we don't have an adult nerve cell, we can't take a skin cell and backtrack it into a nerve to fix spinal cord injuries. That's just not possible. The only way to do that would be to start with those original stem cells. So they're less versatile, and so they can do fewer things. They're less promising for treating diseases. To date, no human disease has ever been cured with embryonic stem cells, at least when your book was published. However, the potential has been demonstrated in mice and rats. So, Parkinson's disease. Parkinson's disease is, they get shaky and stiffness, and the reason for those symptoms is the brain stops making a chemical called dopamine. Well, we've taken rats that have Parkinson's disease, so their brains are not making dopamine, and injected stem cells into their brains and allowed them to be cured of Parkinson's disease. So, was it Michael J. Fox has Parkinson's disease? If it were legal to treat humans the same way we treat these rats, then he could theoretically go in to any hospital, be treated with embryonic stem cells, and within a week or two be free of any symptoms of Parkinson's disease. Now, it's not changing the original DNA genotype, so he still has the disease, and we don't know yet how often he would need to go and get treatments, or if just one treatment would cure him forever. But anybody with Parkinson's disease, we could cure them, or at least remove all of their symptoms, but that's not ethically allowed because it uses embryonic stem cells. Also, multiple sclerosis, that destroys the myelin that wraps nerves, and we've shown, using a similar experiment to Parkinson's disease in mice, we can inject embryonic stem cells into the area and they will produce myelin and cure the mice of multiple sclerosis. Again, this hasn't been done yet in humans just because it uses embryonic stem cells, and there's a lot of controversy surrounding those. Is it okay to use embryonic stem cells, or is it not okay? So where does a research institution get embryonic stem cells? Well, in the past, embryonic stem cells came from fertility clinics. So women would go in, and this is if they're doing in vitro fertilization, they would donate their eggs, somebody else would donate sperm, they would be combined in a petri dish, and usually you collect as many eggs as possible, 30 eggs maybe out of the mother when you collect the eggs for in vitro fertilization, because the mother has to take a long course of shots, so she's injecting herself with hormones every day. It's an invasive surgery to go in there and collect the eggs to combine them in the petri dish and produce all of the little blastocyst embryos. And then you select only some of those embryos to put back into the mother to carry and produce offspring. The other embryos, in the past, they had three options. They could choose to keep them for their own future use, and they would pay a fee for storage. They could donate them to other mothers who couldn't even produce their own eggs, so basically allow other women to have offspring, or they could give them to research. So full informed consent allow agencies to do embryonic stem cell research on them. However, in 2001, the government said, well, this is illegal. You can't do that anymore. You can either keep, donate, or I guess the fourth option that was always out there, you can trash them. So the eggs that are not kept or donated, they're actually just incinerated, so burnt up as medical waste and thrown in the trash. That's what the fourth option is. As of 2001, you can no longer do research. Just keep, donate, or trash. And the laws have changed a bit since then, so this is a little bit out of date. In 2001, in addition to removing that option, they also said, well, now you can only work with pre-existing cell lines. You can only use the stem cells that you currently have. Well, to keep them alive and to keep them stem cells and not let them develop any further, they had to fuse them with mouse cells, just to provide nutrition and keep them alive as stem cells, because they don't want to grow them any further than stem cells. So now you can do research on them and you can demonstrate the benefits of stem cells, but because they've been fused with a mouse cell, these can no longer ever be used in humans. So it prevented any human use of embryonic stem cells, with some exception. Great Britain, Harvard privately funded, Stanford privately funded, and California privately funded has continued to do some work with stem cells, but again, they can't collect new stem cells, so they're all fused with mouse embryos just to keep them alive. So any entity that receives government funding simply cannot work with stem cells. My question is, you guys have probably already noticed this gal over here, I think Nadia was her name, and she had gone through the in vitro fertilization because she couldn't have children naturally, and she had already had some children, and she went back to her doctor, and the doctor said, all right, we'll give it one more try. You're running out of eggs. So they put some developing embryos into her, and none of them took. None of them worked. We went back to her doctor one last time. Her doctor said, all right, you're almost too old to do this procedure. You have, I think she had like 4 eggs left in the freezers. Would you like to try all 4? Because our last treatment didn't work. This is it. So she tried all 4. She never planned on getting 8 babies. Problem was, some of them twinned. They split spontaneously and produced twins, so she ended up with 8 offspring. And I always thought it was interesting, on the news, the same politicians, so politics, the same politicians that were just saying awful things about this young woman because she was a single mother, and granted she has a few issues, but being a single mother and choosing to have that many children, those were the same politicians that are very outspoken against embryonic stem cells. So I would have thought that they would have been in full support of her because she used every single one of her eggs and gave it a chance to make a baby instead of throwing them in the trash. So kind of wherever you stand on that, that's perfectly fine, but that's kind of how embryonic stem cells are used. Rejection of them could be a problem. If you receive somebody else's embryonic stem cells, its DNA is not the same as yours, so your immune system may attack it, could reject it. So it's very similar to an organ transplant, could have problems. So something else called therapeutic cloning has been developed, where you take a nucleus from the adult that's being treated, take an egg that's had the nucleus removed, so anucleate, they remove the nucleus, and then you grow that into stem cells and use those stem cells to treat the adult. And the ethical issues surrounding that, that process, taking an adult cell, fusing it with an egg cell without a nucleus and growing it, should sound familiar. Basically, those would be the first steps in human cloning. That's exactly how we got Dolly. Taking an udder cell and fusing it with an egg cell and growing a baby. So those are the first steps in human cloning. So how far do we allow that to go? Do we stop it at the blastocyst stage, somewhere along here? Some things we can't fix with embryonic stem cells. If you have a heart problem or a liver problem or a lung problem, those are complex organs. We don't know yet how to make those embryonic stem cells grow into those full organs. So can we grow a clone of you, grow it up further past the embryonic stage so that we can later harvest those organs? That is obviously unethical on many levels, but how do we ensure that that wouldn't happen? Or perhaps for myself, I would be appalled at ever doing that. But if something happened to one of my children and they needed an organ and that was the only way to get it, we could grow a clone, I don't know. That would be pretty hard to just let my children go if there was a way to fix them. So there's a lot of ethical issues, huge ethical issues surrounding that.